Okay, today's topic is walking the audience through how penicillin can be used to kill bacteria, mostly focusing on cell permeability and then describing how the bacteria has enabled to prevent this focus on cell permeability, and following using the program Chimera show the bound structure of penicillin. Please cover the enzymatic inhibition mechanism of penicillin and also use images and show the PDB coordinates. Okay, well, start off by showing that you can see the structure of PBP, a acyl enzyme complex with penicillin G. As you can see, let's rotate this around. The structure, the symmetry, there's none, and yeah, that is the structure. As I can spin it around. Okay, next we're gonna start discussing penicillin. Penicillin is an antibiotic that is used to treat and cure bacterial infections. It acts as an inhibitor and binds to the active site of the bacterial enzyme. This enzyme uses the bacteria to synthesize its cell walls, and then the penicillin binds to the active site of the bacterial enzyme. The enzyme loses its activity, which causes the bacterial cell wall which causes the bacterial cell wall to which it affects the formation survival of the bacteria itself. It is mainly important to note that the role of penicillin does not affect the human cells, as these cells do not have a cell wall, which means that the penicillin helps to cure the bacterial infection by destroying the bacterial cells without affecting humans. Now that's done, the mechanisms of how penicillin is used to killing, a mechanism of killing, which is called the beta-lactam antibiotics inhibit the transpeptidases so that cross-linking, which maintains the close-knit structure of the cell, does not take place. The enzymes and related proteins constituent the penicillin binding proteins, which are called PBPs, have been located in the bacterial cell membrane. Each organism has several PBPs, and the PBPs obtained from different species differ in their affinity towards different beta-lactam antibiotics. When a bacteria is susceptible, it divides in the presence of an antibiotic. This term called CWD, which is called cell wall deficient, forms are produced. Because the interior of the bacterium is hypersomatic, the CWD, which are called cell wall deficient, swells up, and then bursts. This causes the bacterial lysis to occur. And then this is how penicillin basically works, or an overview. And the types of bacteria that are susceptible to penicillin are PNG, which is a narrow-spectrum antibiotic, which its activity is limited primarily to gram-positive bacteria. And then there are a few gram-negative ones, and negative ones that are anaerobes as well. Cocci, streptococci, except viridins or enterococci, are highly sensitive, so that means that many of the monococci are many of the monococci. And then some that are gram-negative cocci are Neisseria, gonorrhea, and North meningitides, which are susceptible bacilli. Gram-positive bacilli are a majority of B. anthracis, Corynebacterium dipteri, and then all of the tetna / Clostridium. Also, I want to show how, this is an example of the mechanism of penicillin inhibition of transpeptidase. The structure shows that as penicillin, the enzyme transpeptidase and how penicillin is used in the enzyme. And then, that's about it. And also, you could say that right here when it states that beta acts mainly by blocking the synthesis of peptidoglycan, which I stated earlier, and how a component of the cell wall formed of pyroglycus strands are cross-linked peptides. And as I mentioned earlier, it also says that it's irreversible, inhibited by beta-lactams. And just a few points I want to say is they also attack multiple targets, which are called PBP, penicillin binding proteins, of which transpeptidase is the main example. And that's really it.